All right, so these are all important molecules. And that's also, we get these molecules being made through the citric acid cycle. All right, so let's look at this a little bit. So when you're done with a round of the TCA cycle, basically all the carbons from glucose have been totally broken down, CO2 given off, but that's good. You want molecules to get broken down to CO2. Then we're going to see that the NADH and FADH2 that were made need to do their job. They haven't done their job yet. And they've been synthesized and now they have to go do what they need to do. And that is get their electrons donated off and get an electron transport chain reaction going. So the next step we need to talk about is called oxidative phosphorylation. Because this is where the big payoff will really come for ATP production. Okay, so the TCA cycle gets you a little bit of energy, gets the glucose or the pyruvate all the way broken down, and then you get these reducing molecules made. Now they have to do their thing. So when I say the word oxidative, what kind of molecule do you think of when I say oxidative? Just me? Yeah, oxygen, don't you kind of think about oxygen, oxidative oxygen, which is legit if you are thinking about like how we generate our ATP, oxygen's involved. So, and then if you take like human fizz, animal fizz, they tend to really focus on the idea that oxidative phosphorylation involves oxygen as the terminal electron acceptor. And it's true in humans and animals, but I really need you to come into the mind of a microbe now for what we're going to talk about next, okay? Because yes, some microbes do something similar to what we do. I mean, as we know, mitochondria were once free living microbes. So yes, however, there's a bunch of microbes that do oxidative phosphorylation, but they don't use oxygen as the terminal electron acceptor. And so I've seen in the past, students get so locked into how they learn about it in animal or human phys that they forget that that's not exactly accurate when you get into the microbial world. So let's go through this. All right, so with animals like us, yes, we use oxygen as the terminal electron acceptor, but oxidative phosphorylation, that term oxidative really does apply more to the idea that you're stripping electrons off of something. You're oxidizing something. Okay, so don't equate it with oxygen. So really, in this class, I prefer that you think about oxidative phosphorylation as electron transport linked phosphorylation. Just think about it that way. With the idea that you're going to get electrons stripped off of one molecule, donated to another molecule. Okay, so we're going to see that it is an electron transport chain process. And through that process, you end up making ATP. So oxidative in this class is really referring to electrons being lost from one molecule and being given to another. Which is technically, when you think about that process, that's a redox reaction. RXN is short for reaction. Okay, so we're thinking about redox reaction. So we are busy right now in our mitochondria. We have oxygen sitting at the end of an electron transport chain process. So it's the terminal or final electron acceptor. Because it's oxygen as the terminal acceptor, we get a lot of ATP from that. we get more ATP using oxygen than any other molecule, that if we stuck something else besides oxygen there, it wouldn't do as good of a job. We use mitochondria for that, but remember, mitochondria were once free-living microbes. Obviously, microbes don't have mitochondria, but they can still do something very similar to what we do in terms of aerobic respiration, where they can use oxygen as the terminal electron acceptor. So where would they do that? If they don't have mitochondria inside, where are they sticking all of their electron transport chain players? Where are they putting it? On the cell membrane. You got it. Okay. So this is quite a bit of the cell membrane. If you're an aerobic bacteria, you want to have this fully up and running in your cell membrane. So now there's a lot of microbes who, because of where they evolved, what habitat they've adapted to, they don't use oxygen, either because it's not available or maybe it's toxic to them and they can't survive in the presence of it. You'll learn about that next week. We're going to get into, in lab, looking at oxygen requirements for microbes and figuring out if they like oxygen or not, if they use it or not. So in a microbe, we zero in on the cell membrane for this because they don't have mitochondria. And in this electron transport chain process, you're going to see if there's electrons moving from donor to acceptor, donor to, I think of a hot potato game where you're just passing these electrons down the chain. And what you have to appreciate is that as these electrons are going from player to player in the electron transport chain, there's a little bit of energy at key points left over or available to pump protons across the membrane. So a fancy way to say that is they conserve some of that energy released during the electron transfer and use it to drive ATP synthesis. So hidden in that little phrase is the idea that in key steps of the electron transport chain, there's a little bit of free energy available to pump protons across the membrane. That sets up your proton gradient, which then is going to allow them to make some ATP. All right, so we're not going to get into huge details of the electron transport chain process. However, there's some key things to know. So this is a kind of the standard example they give in a micro class. In this case, it's aerobic respiration because you see oxygen is the terminal electron acceptor. So that's where the game stops. And you can see that when they accept electrons, the oxygen will get reduced to water, which they don't really, care about. They just kind of release that as waste unless they need it because they're dehydrated. So here you have a couple of things going on. You see NADH up top. So there's one Tinkerbell donating off electrons. The pixie dust. Here's the other one, FADH2. Okay. So at complex 1 and complex 2, you can see they have them all, but that's typical. Not all pictures have that, but we number them complex 1, 2, 3, and 4. So you can see that NADH is donating off electrons. If you follow the black line here, you can see there's electrons moving from donor to acceptor. FADH2 is also donating electrons. So those are the ones coming from the citric acid cycle. All right, so they wandered over to the membrane and said, here, you need some electrons to get going, go ahead. And you can see that there's many players in the game that are passing on the electrons on down the line until you end up with oxygen getting those electrons. Oxygen actually has to get 4 electrons in quick succession to form water. And then the other thing that you have to appreciate is that as all of this is happening at key points, you have enough free energy to pump protons across the membrane. Well, really, no matter what you're talking about, an animal, including us, or a microbe or a plant, they always do this. They evolve this mechanism where they set up an electron transfer chain somewhere. In our case, we're thinking about microbes, so it's the cell membrane. And then they link it to proton pumping. So anytime someone starts talking to you about an electron transport chain, you should start thinking about a proton gradient. That's the goal of the game. There's a couple of goals, but that's a huge goal. I want to create a proton gradient and I want to pass my electrons on, and it like with photosynthesis, when they're doing that in the chloroplast, there's another payoff because their terminal electron acceptor is a really important molecule. But anyways, so here you see this idea that there is this electron flow and it's linked to proton pumping. Okay, electron flow, proton pumping. Questions on this idea? So as this is happening, you get the proton pumping going on. So then you get that lovely proton gradient. You get lots of protons on one side of the membrane, not many comparatively to the other. So it's over here that you see lots of protons building up, and they want to diffuse back to even out their concentration, but they can't go through that hydrophobic portion of the membrane because they're charged. So instead, they'll go through that ATP synthase. I really wish the artist that drew that picture had included the ATP synthase, because this is like the huge goal of why do you get a proton gradient. And so here you see the protons diffusing back through the ATP synthase, and that creates a torque force where it allows these subunits to rotate, and it allows the ADP and inorganic phosphate to come into physical contact and react. Then you get that wonderful ATP. It's really an amazing process. They call the ATP synthase a little molecular motor, which is a good description. So the artist should have drawn that back onto this picture. They should have stuck that at the end here, maybe, but they forgot. Okay. So link the electron transport chain. As electrons are flow, you get proton pumping, which leads to a proton gradient, which then leads to ATP synthesis. Okay, so the NADH and FADH2 that were created in the citric acid cycle wander over to the electron transport chain, donate their electrons in to at complex 1 and 2 of the chain, and you get that. I call it a proton gradient, but maybe in other classes they call it the proton motive force or electrochemical potential. But it definitely is a proton gradient. That's the molecule. And you end up with the ability of the bacteria to make ATP off this. So this one's not in your textbook, but at least the artist bothered to include the ATP synthase as you're looking at kind of a simplified electron transport chain process with oxygen sitting at the end. And you get the proton pumping going on, and then the formation of ATP. Going back to this figure, you see how there's these numbers here, negative 0.32 volts, negative 0.22 volts, and then all the way down here where oxygen sitting is positive 0.82. So this is something that we're going to be talking about soon. This is the idea of the reduction potential, how when you are at a negative value like that, that means that those molecules are very willing to donate off electrons. They have really good energy to do that. If something's on the positive scale, that means they're really good at accepting electrons. Okay, they love to accept. And oxygen is the best electron acceptor on the face of the earth. Okay, oxygen is the best. Hands down. So when you see a negative like this, that means those molecules are very happy, chemically speaking. They're very good at donating off electrons. It's very easy for them to do that. So that's why we call them energy carriers. They're reducing molecules because they can donate off an electron and reduce some other molecule by doing that. So when you have this process going on, I don't know, let me just see if this helps you at all. I can post this if this helps. But when I was first learning this, let's see, where did it go? Well, now it won't let me get to it. Oh boy. Let me get to it this way. All right. Let's try that again. Technology. All right. So if this is a little puzzling, maybe this will help. So as the electrons are traveling through the chain, they go from a higher to a lower energy level, and that's being denoted by these nice negative numbers of when it starts off, and you end up at areas that have a really positive voltage. Okay, so that's referring to that idea. So they're moving from less electron hungry to more electron hungry. So oxygen is very, very electron hungry. It's very, very hungry to get those electrons, whereas NADH and FADH2 are like, I love giving off electrons. Think about Tinkerbell sprinkling that dust everywhere. All right, so the process is downhill. They often refer to it as the energy is released in these downhill electron transfers as you go from negative to positive voltage. And that creates a situation where you have a little bit of extra energy in those key steps to use to pump the protons across the membrane. So I thought of it this way, where I had a family dog. His name was Jerry. And when we would go sledding on Whitman Hill, he would chase after us and want to jump on the sled and come with. And so when I was learning this process, I would think of, well, I'm the electron donor and I'm carrying an electron, the kid in this case. And my dog is the proton trying to grab onto this energy as we're sledding. Right? And so if the dog can jump onto the sled, that's that little bit of free energy now available to get that proton pumped across the membrane as the electron is being passed down the chain. So if the dog is successful, jumps on that sled going downhill fast, okay, then that's the free energy. He's like, all right, I got a free energy. I'm getting a free ride. I'm going to use that and I'll be able to get pumped across the membrane. And so if you can imagine that happening, I picked a dog sled because we're doing the UP 200 soon. But if you can imagine a lot of dogs and a lot of protons ending up now on the bottom of the hill or the other side of the membrane. And so now you have a gradient, a proton gradient. And that can be used as they diffuse back across, that can be used to make ATP. So for some reason, that analogy stuck with me and really kind of made the light bulb come on when I was trying to imagine this process. So if that helps, I'll post it and you can make use of it. But, and then there's a couple of extra cute pictures. So this idea of that potential energy at key spots, because of the way you have it going from a very negative voltage to a very positive voltage as they pass along that electron transport chain. Okay. Questions on this idea? No questions? Can't be crystal clear already. Okay. I really like this picture. But I don't know about you, but when I took biochem, I learned all of this in isolation and it was a lot to learn, right? Because you had to know glycolysis in detail. You had to know step by step by step, all the enzymes and so you used to, like just focus on one at a time. But this one, once you learn how it all fits together, I think that also can help the light bulb come on a little bit. Because you do have linkages between all of these. And so, for example, we talked about how with glycolysis, once you get down to pyruvate, the microbe, if it's smart, should be assessing the environment and saying, if you think about E. coli that loves to grow with oxygen. Well, let's think about E. coli. So E. coli would be like, my top pick would be oxygen. I want to use oxygen. And let's say we're providing a lot of oxygen for it, so they'll, great. I'm gonna shift over to the TCA cycle or citric acid cycle, run that and get my reducing molecules, NADH, FADH2, get a little bit of energy. And then I'm gonna funnel the FADH2 and the NADH into the electron transport chain process. Because I have oxygen available. And so, where's oxygen? Well, here, they're just putting it right here. And so oxygen is going to be the terminal electron acceptor for E. coli. And when that is run to completion, you end up with a lot of ATP. They're showing 34 ATP just through the electron transport chain process. So if E. coli is hanging out in an environment with oxygen, that's its top pick. Oxygen is the best electron acceptor on the face of the earth, which correlates to you get the most ATP if you use oxygen as the terminal electron acceptor. So go for oxygen if you have it. Now, one other thing I want you to keep in mind is that we talk a lot about a proton gradient being used for ATP, which is totally legitimate. However, microbes can do other things with a proton gradient. All right, we already talked about the idea that they can use it to power the rotation of the flagella. They have to use a lot of protons for that. But they also can use it to pump out toxins that they're getting exposed to, like antibiotics. That's one way they can become resistant to antibiotics, is really evolve a mechanism to pump out the drugs. They show the flagellar rotation, getting things brought into their cell or out of their cells through the use of symport or antiport mechanisms that involve protons. So they can do other stuff with a proton gradient. It's not just ATP. So keep that in mind. Okay. So that's the end of the slide set on the intro idea. So we've covered glycolysis, citric acid cycle, and the electron transport chain. So now what I'd like to do is build on that a little bit. and think about what microbes can do that we can't for the most part. All right, so the next thing we got to get into is redox reactions. How many of you have had redox reaction concepts in some other class? Probably a little bit, maybe in Chem 112. If you're in biochem, I'm sure you're getting it. I don't know about you, but when I was first learning this, it was a head scratcher in the sense that they always tell you that electrons are transferred. But when I'm looking at the reactions, I'm like, what, where? And so if you can remember that it's often shown as a transfer of hydrogen, then you're set because you have to remember hydrogen is composed of an electron and a proton. And so you are getting transfer of electrons if you see hydrogen getting involved in the game here. So that's what they're showing here. They're showing the oxidized molecule up top, and then once something gains an electron, it's in the reduced state. And you can see they're showing the addition of the two hydrogens coming in to facilitate that process of oxidation to a reduced molecule. All right. So energy from these redox reactions are necessary for getting a lot of your ATP. When we look at them, we talk about how they occur in half reactions. There's two half reactions that make up the redox reaction in total. So you have an electron donor and an electron acceptor. So the donor is going to be giving off the electrons. And once it does that, it becomes oxidized. The electron acceptor, once it gains that electron, is going to be reduced. Okay. So if you donate off an electron, once you lose that electron, you've been oxidized. Once you gain that electron, you've been reduced. Have you learned it this way? Leo the lion says grr. What's the other one? Oil rig. Yeah. So loss of electrons is oxidation. Gain of electrons is reduction. Or oxidation is loss. Reduction is gain. So whichever way you want to remember it. So let's look at this one. The overall reaction is down at the bottom where you have hydrogen gas reacting with oxygen and you get water. But you can pick it apart into two half reactions. And so up top, if you do the thermodynamic calculations for that, you get a reduction potential. D sub 0 with that little prime number is the notation for reduction potential. And you see it's a negative value. So that means that hydrogen, H2, has a negative reduction potential, which means hydrogen's very happy, chemically speaking, to give off electrons. So when you know that, then you know when you look at the overall reaction down here, you can say, oh, hydrogen is going to be the electron donor. And then if we look at the reduction potential of the other half reaction, you see it's a positive value. So oxygen doesn't want to donate electrons. If it's a positive value for this, the voltage reduction potential, if it's positive, that means that molecule is good at accepting, not good at donating, right? So then we know, oh, okay, well then oxygen is going to be the one accepting electrons in that bottom reaction. And when those two react and do their electron donation and accepting, you end up with water. So you can do this and calculate reduction potentials for all kinds of molecules, and they all vary. It depends on the molecule. They vary in their tendency to be oxidized or reduced, how happy they are to give off electrons, or how stingy they are about it. All of that can be done. And you can calculate all these reduction potentials, and they're either going to be a negative value, 0 or positive, okay? And one thing I want you to remember is that oxygen is an excellent electron acceptor. As far as we know, it's the best on the face of the earth. And you should also appreciate in this class, because microbes love hydrogen gas, H2 is an excellent electron donor. And there's a lot of hydrogen gas on the face of the earth and oceans, so they can make use of this. Okay, your book has this table, which is the so-called electron tower. And it gives you reduction potentials with the kind of half reactions here. To me, that's a little, it doesn't work well with my brain. I prefer this one. So I'm going to teach off of this one. It's presented a little differently. So this is giving both half, well, redox couples. So this is a tower showing redox couples. And by convention, we always have it where the negative numbers are up top and then they get progressively closer to 0. And then you start getting the positive scale as you go all the way to the bottom. So the other thing to be aware of with this is that the redox couples, when you look at them, there's a little slash in between them. So let's look at the H plus slash H2. Okay, so by convention, the oxidized form is to the left of the slash, and the reduced form is to the right of the slash. Okay, so the oxidized version of hydrogen gas are photons. All right. You can see that you have a very negative reduction potential, negative 0.6, and then it starts to get, negative 0.3, 2, 1, and then here's your 0 mark, and then you start getting positive numbers. So everything on the top here in the negative scale means that the partner to the right of the slash, the reduced version of all of these molecules, so H2, glucose, methanol, NADH. So the partner to the right of the slash, when you're up in this region of the tower, that's the molecule that is happy to donate off electrons. And so H2 loves to give off electrons. Glucose does, NADH does, okay? So that's how you interpret what's going on here on that part of the tower. Now it kind of flips now when you go into the positive portion of the tower. So if you look, oxygen's at the very end, and no matter what redox tower you're presented with, I guarantee you, oxygen will be at the end, at the very bottom, all right? So you see oxygen slash water. So now the way you interpret it down here in the positive is that anything to the left of the slash is going to be accepting those electrons. Okay, so oxygen loves to accept electrons. It's to the left of that slash. What do you think? Kind of makes sense? So that's kind of the rules of the game here for how to interpret when it's presented this way. So then they have these different arrows here. What they're trying to show is, let's take and make some reactions. A few, 3 different ways of combining these molecules into a reaction. So I think we're going to start with that on Monday. We'll stop here. The next thing I want to do is get into EDUCAT. I have the essay scores. I have the essay portion graded, and so I can let you into that and you can look through your exam, the essay portion. And then we will review